Hello, everyone, and welcome to today's webinar titled Streamlining Cell and Ex Vivo Gene Therapy Manufacturing, Innovative Solutions from GenScript. This webinar is a part of the 6th annual event in the Bioprocessing Virtual Event Series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I'd like to now present today's speaker, Daksha Patel, Senior Segment Marketing Manager, Cell Therapy at GenScript USA. For a complete biography on our speaker, please visit the Presenters tab from the menu on the left of your screen. Daksha, you may now begin your presentation.
Hi, thank you for a nice introduction, and hello everyone, welcome to today's webinar session. In this session, we will explore a brief overview of the challenges faced by the cell and ex vivo gene therapy developer and how the GenScript can support you in streamlining your process development and the commercialization effort. Here is an overview of what we will cover in this webinar. I will start with introduction to our company, and following that, we'll introduce different stages of cell therapy development and highlight the key service and product needed for each stage and their importance. Next, I will share detail about the GenScript strength and capability and how GenScript can support you during your cell and gene therapy development journey. Let me start by sharing some background information about the company. GenScript is a biotechnology company which provides the life science service and product worldwide. And key milestone of the company's success are shown here. In 2002, we launched the first custom gene synthesis platform. And 2013 launched Bestzyme, which is a subsidiary company for synthetic biology product. 2014, we launched the Legion Biotech, which is another subsidiary company specialized in cell therapy. And 2019 launched GenScript ProBio division, which is a biologic CDMO service provider. And 2022, Legion CAR T therapy received US FDA approval. So in summary, GenScript has a long-standing record of innovation and excellence in the life science, and it's well known for one of the top service provider for the in the field of cell and gene therapy. Our mission is to provide innovative technology and solution worldwide that boost advancement in the life science research, biologic development, and cellular therapy. Ultimately, our goal is to enhance the human health and well-being. GenScript headquarters is located in New Jersey, Piscataway, and with additional research and manufacturing sites are also located in China. In 2021, we opened the new life science facility in Seattle, US, Singapore, and Shanghai. Additionally, we have regional offices in the Korea, Japan, Europe. And as a result, we empower the researcher, clinician, and biotechnology company with our cutting-edge technology and services, ultimately which helping them to do scientific research and transferring that discovery into benefit of their society. GenScript has been referenced in numerous peer-review journals, and GenScript has made significant contribution to the scientific community by facilitating the publication of over 87,000 research papers in reputed journal. And this publication demonstrate the impact of our product and service on advancing scientific knowledge and how we support the researcher worldwide. After introducing the GenScript, their mission, and their value, in this section, I will provide an overview of the cell therapy and discuss some common challenges in the CGT field. So typically cell therapy development process has four stages which include the target discovery, lead generation and optimization, preclinical development and clinical development and manufacturing. At discovery stage, scientists aim to identify the disease target and evaluate the cell types that could address the medical condition along with the therapeutic effect. Subsequently, in the lead optimization stage, optimizing CAR design and developing cell engineering technique are crucial to enhance both effectiveness and cytotoxicity. Preclinical study are then conduct using more complex animal model to evaluate the therapy safety, efficacy, and potential side effect. And finally, numerous cell therapy can progress to clinical trial where their safety and efficacy are tested in human patients. If clinical trials are successful, the therapy may eventually receive the regulatory approval and become a mainstream treatment for various disease and medical condition. Finally, with successful validation and clinical trial, many cell and gene therapy require the GMP production. As noted in the previous slide, the discovery of cell therapy involves numerous crucial steps, such as target identification and validation, CAR optimization, and many others. And each of these steps can be addressed using the various tools. For example, at the discovery stage, target genes are often validated using CRISPR for gain or loss function study to understand the target gene function in biology and disease. To facilitate discovery effort, GenScript provides a wide range of cutting-edge technology and services. Here, few of them are represented, like which including the molecular biology, gene editing, protein, mRNA, oligo, peptide, antibody, etc. So it's provide you one-stop solution to accelerate your research. So after the completion of target and lead optimization, at the development stage, scientists start considering the manufacturing of cell therapy. And the standard manufacturing process for CAR T cell therapy includes it's a cell isolation, and cells are isolated from patients if it's autologous cell therapy, and it's from the healthy donor if it's allogenic cell therapy. And then cell activation and selected steps are also one of the key steps. And another very important step is a cell engineering or genetic modification of the desired cell, where selected cells are modified to enhance anti-cancer property using the viral or non-viral platform. And throughout all this step, various quality control measures, safety assessment, and regulatory compliance check are performed to ensure the safety and efficacy of the cell therapy. So cell therapy manufacturing process is continuously refined to increase the purity, potency, stability, and reproducibility and batch size, while also the developer is trying to cut down on time and the cost. So successful validation in clinical trial often require the GMP production for the cell and gene therapy, and cell therapy manufacturing is a complex process as we discussed earlier. It involves multiple products from various sources, including ancillary raw material and non-conventional raw material. And this complexity impact their safety, reproducibility, cost, scalability, and quality consistency. Obtaining essential material is the major challenge, and in this disruption in the supply chain can cause significant production delay also. So selecting the reliable reagent supplier is critical to prevent the GMP complication, And to address this challenge, GenScript provide top quality RU and GMP grade reagent and service with supply chain guarantee. We offer phase appropriate reagent, customizable solution, and regulatory compliance support and robust automation capability to avoid therapeutic development. To advance your therapeutic development. In this section, I will explore how GenScript can support or effectively add in the three critical steps of the cell therapy process: isolation, activation, and expansion. And I will walk through some tools needed for each stage and their importance and GenScript capability and the strength. So, GenScript has developed cytosine technology to support the cell therapy field. This technology is particularly help in ex vivo cell therapy, where the isolation of desired cell or depletion of unwanted cell is necessary. To facilitate this, GenScript offer antibody coupled magnetic bead, and all the key ingredients used for developing these beads is fully produced in-house, and including the antibody and the magnetic bead. And these ingredients have undergone strict validation within the R&D and with the beta tester to ensure their quality and effectiveness. So the cytosine cell separation solution offer a comprehensive toolkit consisting of antigen-specific paramagnetic nanobit and automation equipment. And this powerful combination provide researcher in the field of cell therapy or ex vivo gene therapy reliable and efficient solution for isolation process. Here I show how the cytosine can be work in the modular workflow, and the reagents are which provide is also sterilized, non-toxic, user-friendly, and available in both RU and GMP grade. They are suitable for use with fresh or frozen PBMC or leukapheresis and enabling with depletion or enrichment process. And nanobeads, which use it's during the cell isolation, assuring the compatibility with various downstream application such as cell culture, activation, and expansion, even including the flow cytometry analysis. For cell isolation, GenScript also offer this cytosine 1000 automation equipment, which coming with the GMP compliance tubing sets, and it's easy to operate and equip with user-friendly interface, barcode scanning for reagents, and it's allow the customizable programming also. Moreover, it's efficiently integrate with the other instruments and make the modular workflow uh, flexible. And let's see the video which demonstrating how the cytosine 1000 instrument um, um, work into the lab. So we watch in the video how the cytosine 1000 instrument is working and how effective it's for the positive and negative selection. Now we are talking about the manufacturing of allogenic CAR T cell, where off-shell allogenic CAR T cell development has many challenges. One of them is allogenic CAR T cell may cause life-threatening graft versus host disease, and the alpha beta T cell are considered as a primary responsible for causing this disease. Therefore, allogenic CAR T cell developer address this issue by different technique, and one of them is removal of alpha beta TCR positive cell using the immunomagnetic separation technology. To facilitating TCR alpha beta depletion or selection, GenScript offer the cytosine TCR alpha beta nanobit. Currently, reagents available in the market for supporting TCR alpha beta depletion typically involve the indirect labeling. So, in which first they're doing the labeling with the antibody, and then they in intermediate step include the washing, and then they're going to label with the bead and then wash again. So the advantage of direct labeling, it can reduce your steps and make the process smooth and reduce the time and streamline in one step process. So it's easy for automation and decrease the risk of contamination also. Uh, and now we are talking about the activation. So, investigation in adaptive cell therapy and CAR T cell therapy have consistently shown that the infusing the less differentiated T cell subset for the cancer treatment lead to improved therapeutic outcome, and therefore achieving the less differentiated T cell, such as a naive or stem cell memory T cell using the activation step, is crucial. And in this experiment, T cell were initially enriched using the cytosine CD4 and CD8 nanobit, and subsequently they were activated using the GenScript ANSID T cell activation reagent, and which was compared to the competitor product also. So, as we show in the result, GenScript and SID T cell activation reagent provide high-fold expansion while maintaining the stable memory phenotypes and ensuring the enhanced downstream application by preserving the high cell viability. And this ready-to-use reagent consists of the CD3, anti-CD3, anti-CD28 couple nanobit, and in this um, um, bid also get the approval. Recently we get the drug submitted for the drug master file, and register number is also available. So this is completely working well in clinical and manufacturing background. And to advance the development of the cell therapy industry and expedite the process of cell isolation, GenScript entered into strategic collaboration with this Persona Gene, the company which doing the cell therapy work. And this collaboration is aimed to accelerate the progress of cell therapy, ultimately benefiting the larger number of patients. And so our reagents are well accepted in the industry, and GenScript also entered into the strategic collaboration with Applied Cell to deliver the combined cell isolation solution for cell therapy drug development worldwide. So, under this collaboration, GenScript will develop and supply our GMP grade cytosine reagent to use the developing CAR T and other cell therapy product on Applied Cell Mars platform, which is I shown here. So these reagents are compatible with different platform. At the same time, many other companies are using these reagents in their cell therapy manufacturing space. Uh, So another very important reagents or key ingredients is used into the cell therapy process after the activation or during the activation, the expansion of the cell. So recombinant protein includes cytokines and growth factors are very necessary material which is used during the expansion or activation. So example in CAR T cell therapy, T cell required the growth factor to expand and which are provided by the serum. One growth factor is a IL-2, which upon binding T cell receptor promote the polyclonal expansion of the T cell. So even if these recombinant proteins are not present in the final product, but the quality can impact the stability, safety, potency, and purity of the final product. GenScript is providing GMP platform to convert the REO grade protein to GMP grade, and these are suitable for use in preclinical and clinical stage. And we are providing the different kind of cytokines for all different type of cells to make your journey more easy in any area of the cell therapy you are working. So in the cell therapy, after doing cell isolation, activation, expansion, the very important one step is a cell engineering or modification. And the effectiveness and safety of CAR T cell therapy can be improved by using engineering strategy, where genetic material is delivered to cell through the vector, which can be either viral vector or non-viral. Each type of vector offering distinct method of introducing genetic material into the cell. Choosing viral or non-viral delivery strategy is an important step in the therapeutic design process. To support this research, various tools are required to facilitate the discovery and development work. So in this section, I will share GenScript capability to support both viral and non-viral cell engineering approach. So before going that, I would like to share the blueprint. Here I shared the blueprint of the car design, where car have four main part. One is antigen binding domain, second is a heme, third is a transmembrane domain, and the fourth is an intracellular domain. Each part has its own job, and researcher can customize the car by adjusting this component. Researchers are using engineering strategies such as gene modification, editing, manipulating gene insertion, or deletion to fine-tune its functionality. For that, they need specific tools and reagents to successfully develop therapeutic cells to make more effective, targeted, and safe therapy. For example, both autologous and allogenic products require specific type of gene addition and deletion tailored to the final products and its intended therapeutic application. So the advancement of CRISPR genome editing has opened up the new era for possibility in the field of precision medicine. And currently, CRISPRs are used in number of clinical trials, and they are under investigation for different diseases, including blood disorder or cancer, diabetic, infection disease, inflammatory disease. So CRISPR-based editing in human T cell can be achieved by both viral or non-viral method. And in this part, I will share what such GenScript can support in this area. So CRISPR-Cas technology is also useful in target discovery or gene function study or genetic interaction and protein testing. So GenScript offer variety of reagents and service for CRISPR-based discovery and research, including the sgRNA design tool, HDR knock-in design tool, CRISPR gRNA plasmid library, even knock-in optimization kit, CRISPR cell line. And this ready-to-use tool help researcher in different parts of their CRISPR experiment. So one of them is bioinformatic tools, and this is a free online bioinformatic tool, help the designing the single gRNA and gRNA library with our advanced algorithm, user-friendly interface, and designing gRNA take just a few clicks, making it easy for user. User can enter their target gene and choose appropriate HDR template and select the desired sgRNA template. They can optimize the sgRNA by adding tag or purifying option or localizing the target protein, etc. Additionally, user can specify the quantity, purification method, and delivery format. So this is a very basic tools which is always needed, and it's available free online. Uh, and Another one is ready to use CRISPR gRNA Cas9 plasmid, which facilitating the precise gene editing, cell engineering, or enhance the therapeutic property of immune cell. So our offering include the CRISPR plasmid, and which is developed by pioneering in Feng Zheng laboratory at the Broad Institute, which are well-validated platform for expressing the CRISPR-Cas9. And GenScript maintain the collection of pre-validated, ready-to-use plasmid that can be conveniently searched by gene name or symbol or ID in our gRNA database. And this support makes genome editing more accessible than ever before. And GenScript also provide the CRISPR gRNA library option mm, from the Broad Institute, and these include the gene knockout library for human and mouse, including the CRISPR SAM library for activating gene in both human and mouse. Additionally, fully customized gRNA library are available for various application, and including like gene silencing or gene wide screening, or maybe knockout or activation. Furthermore, our CRISPR gRNA library construction service utilize the oligopool synthesis technology, so effectively that can reduce the cost and accelerate the gene target screening timeline. In the field of cell and gene therapy, CRISPR knockout and knock-in cell line is an easy and efficient tool for target discovery, drug screening, disease biology study, or signaling pathway study. And to generate effective CRISPR-mediated gene knock-in or knockout cell line involves a sustainable workload. So include the designing the gRNA template, and here I show the construction process for the CRISPR cell line, which include the um, design and synthesis of the gRNA, transfect the cell line, then analyze editing and knockout efficiency, and conduct the assay. So to the customer who do not want to do whole cell line development in the lab, and to speed up their discovery and validation work, GenScript offer the different CRISPR gene editing cell line by using the chemically synthesized a single standard gRNA. We employ RNP delivery system with high editing efficiency and which gives low toxicity. And we also support the various delivery method like viral vector or plasmid, and gives the customer to select whatever they method they want. At the same time, customer can specify the target gene, editing region, cell type, etc. And in addition, customization service for the QC analysis also available along with the regular service for the CRISPR cell line. Um, CRISPR-based gene editing relies on the Cas9 enzyme, which is acting as a molecular scissor. As a CRISPR gene editing application transition from basic research to clinical manufacturing, the need for high quality raw material and comprehensive uh, complete documentation become increasingly crucial. To meet this requirement, GenScript provide the validated GMP grade and RUO grade Cas9, Ultra Cas9, and ESP Cas9 protein. We also providing the Cas 13 and 12 nucleus, and this nucleus follow good manufacturing practices and ensuring that aseptic manufacturing and all the QC testings are provided along with the GMP grade reagents. For CRISPR-based gene editing applications, smartly designed sgRNA survey serve as a guide for directing the Cas9 protein. And combining sgRNA with Cas9, which make the RNP complex, offer the additional advantage over the traditional plasmid or virus-based delivery system. So, GenCRISPR synthetic sgRNA are proven as effective for targeted gene editing in knockout and HDR mediated knock-in experiments. We are providing two type of sgRNA. One is a easy edit sgRNA, second one is a safe edit sgRNA. Easy edit sgRNA is a more economic solution, so high editing efficiency and flexible at scale. And safe edit sgRNA is HPLC purified, can help to reduce off targeting and show minimal impact on the cell viability. Additionally, GenScript also provide all add-on QC optional like microplasma testing, endotoxin testing, um, cytotoxic testing, stability testing, etc. And we also offer the option delivery 96 well plate for facilitating high throughput screening in the beginning stage. Uh, One powerful application of CRISPR-Cas genome editing technology which involved the precisely inserting DNA templates through HDR pathway. And selecting the suitable HDR template is a very critical. So GenScript offered the high quality sequence verified HDR template both in the linear format as well as in circular format. One option is a single standard DNA, which support the lowest cytotoxicity for the highest efficiency and minimize the off-target efficiency, making it ideal for T-cell therapy and cell and animal model generation. The second option is a double standard DNA, which covalently close end of linear DNA enhance the stability and reduce the risk of non-homologous end joining, and therefore it's improving the accuracy of knock-in also. It's also ideal for large-scale screening and process optimization and can be used in cell and gene therapy. And the third option which we're providing is a gene circle dsDNA. It's a novel small circular double stranded DNA vector with no antibiotic resistance gene, and it serves as an efficient knock-in template with lower cytotoxicity and ideal replacement for plasmid and HDR template. And it can be utilized in various applications such as knock-in template or virus packaging and non-viral vector. So now I will share some case study or successful customer cases where they use this CRISPR technology in their day-to-day experiments or in the cell therapy development. So here represented the GenScript and Gladstone UCSF Institute, which they uh, mm, published one joint publication in the Nature Biotechnology, which describing the method for achieving 40% plus knock-in using the GenScript non-viral ssDNA in primary human T cell. So in this case study, Dr. Brian from UCSF developed the hybrid ssDNA with Cas9 target site as a novel payload for non-viral method to genome editing. And subsequently, they developed the GMP compatible workflow using the fully non-viral method for improved knock-in efficiency. Few data of that experiments are shown here. One of this figure, which is a diagram of non-viral CAR T cell manufacturing process they used, and the upper part of this three figure, which shows that GenScript SSCTC consistently perform better compared to the lab generated SSCTC or standalone ssDNA. And then in the bottom two, They shows the knock-in efficiency, and where GenScript SSCTC via electroporation without any enhance can reach 46% knock-in at the clinical scale production. And the last two graph is showing the knock-in positive live cell count generated by GenScript SSCTC was above estimated patient dose, and in vitro assay demonstrated efficient killing of PCMA CAR cell. Another case is like Neogen, which is a biotech company and working on TCR therapy. And the company developed the manufacturing and commercialization portfolio for TCR product, including autologous and allogenic. So they develop fully individual, personalized neoantigen specific TCR therapy with novel T cell engineering approach, and they able to efficiently select and tracking TCR edited T cell that will be utilized for T cell product manufacturing. So, to apply the standardizing TCR gene editing efficiency in clinical application, for that they decide to sourcing the GMP grade DNA repair template from GenScript, and they adopt the covalently close and linear double stranded DNA template. So when editing T cell, typically they use 1 microgram DNA template for both with and without MTX. The graphs are shown here, and editing efficiency reach 80%. But however, to reduce the cell viability issue, the DNA dose is reduced to 0.25 microgram as shown in this graph, and at this reduction dose also TCR editing efficiency remain 80% and the cell viability remains significantly higher. So GenScript dsDNA, which is close-ended, help them to develop the TCR-T personalized cell therapy. Uh, Another case study is with Editas Medicine, which is a leading gene editing company using the CRISPR gene editing technology. They developed the SELECT technology, and uh, initially they developed the SLAC combination with AAV, but due to the known manufacturing difficulty linked to AAV and similar viral vector, they tried to investigate whether the Slack could facilitate the effective knock-in using the non-viral DNA template. Um, for figure is like here they shows how they did testing of non-viral DNA template with Sleek, and for that they optimized the cell protocol for linear dsDNA and ssDNA template. Oh, sorry. Upon comparing the Sleek knock-in efficiency, day 7, viability, and fold expansion among these three various DNA templates, they observed minimal difference between the ssDNA template and AAV6. However, linear double standard dsDNA displays slightly lower viability and expansion on day 7 compared to the single standard ssDNA and AAV6, which we can see in this graph. And for this close-ended dsDNA linear template, which were they contacted in GenScript and which is generated by GenScript. So further, the another Team Exum is a biotech company which also developing the UCAR-T, and GenScript Biotech and Team Exum Biotech make the strategic collaboration where GenScript will provide the Team Maximum Biotech with a various CRISPR reagent to support their development of UCAR-T product from discovery to commercialization. And we also make kind of a cooperation agreement with the Biohang to develop and produce the key raw material for gene and cell therapy. So not only that we're providing the CRISPR gene editing tools or CRISPR service and reagent to the different company, but we also make collaboration with the Evicta, which they have like mm, Solupor platform, which is automated non-viral delivery system designed to facilitate the clinical manufacturing of advanced cell therapy within a controlled environment. And this platform offers versatile delivery of diverse gene editing tools such as mRNA, CRISPRs, or plasmid. And so GenScript and the Evictia team up to improve the non-viral cell therapy manufacturing process. So in this partnership, all the uh, this partnership helped the commercialization of non-viral cell manufacturing process into GMP compliant system quickly. So GenScript provide RUO to GMP grade services and reagent for the mm, non-viral gene and cell engineering from research to clinical phase. And we offer all the CRISPR reagents and service in the stage appropriate format. So if customer working in the discovery stage, they can use the REO grade reagents and slowly they can go with the GMP like and GMP sgRNA. And along with each the stage, we are providing different QC specs to support their successful IND filing. Since 2020, GenScript have been implementing a phase-appropriate GMP capability, and this plan include the establish the GMP compliant facility, including the GMP compliance management and personal training. As a result, our CGMP Nanjing site now has a 5 manufacturing line, and its response to the clinical progress of our client and increasing demand for GMP grade CRISPR material. We also constructed GMP facility in Jensing sites, which has the 12 manufacturing line. So GMP capability is we enhance and be able to produce the sgRNA from 100 milligram to the gram with capacity of 400 batches per year. And additionally, we can produce the ssDNA and dsDNA to milligram to the gram level and capacity to 100 batches per year. And our integrated quality management system also support the IND filing, and we are providing like 10 different QC options to meet the global IND filing requirements. So our comprehensive phase-specific CRISPR gene editing solution offer flexibility to support your project any stage and with the compliance of the FDA, EMA, or NMPA regulation. And production facility has the key feature like it has a GMP product environment, clean chutes and sterilized filtration, QA, CQA and CPP conform, etc. And at the same time, we are providing the QA and QC documents, which is including all the batch record, identity, purity, sterility, and CGMP compliant material, supplier management, etc., including the validated QC testing procedure also included. So this is like we trying our best to support the cell therapy and gene therapy developer to help their commercialization activity. So, with 20 years of expertise in nucleic acid synthesis, GenScript has developed and optimized the process, uh, quality control, and extensive experience in establishing um, mm, uh, CGMP level CRISPR nucleic acid reagent product line. So, since 2020, we have support so many clients for their clinical project, and GMP grade CRISPR's material also deliver. And I highlighted few numbers here. So we are able to deliver more than 100 batch for the clinical grade of this CRISPR reagent, and we able to provide like 7 IND filing on the globe, and also did 30 IND filing, and 22 successful audits. So, in conclusion, GenScript is trustworthy partner. And if you are developing the cell and gene therapy with non-viral cell engineering, definitely you can rely on GenScript service and product to bring your product in market as early as possible. So, in this section, because in recent year, a growing number of researchers have been exploring the use of IVT mRNA for developing T cell based immunotherapy, I will show what GenScript can help you in this area. So, since IVT mRNA leaves no ultimate genetic residue in the recipient cell, it's offer much safer CAR T cell therapy compared to the viral CARs. And mRNA can use for both reprogramming of T cell as well as gene editing with Cas9 family of nucleus. So manufacturing of mRNA CAR T cell in which encapsulated mRNA molecule are taken up by the T cell and act as a template for the production of targeting receptor and effectively reprogramming T cell to attack the cancer. So here I just shown the few figure how this mRNA based cell therapy works. And Cas9 mRNA is a versatile tool for precision gene editing and has wide range of application. So this mRNA is capping with cap1 structure to enhance the translation and designed with 100A tail to mimic a mature mRNA. And the sequence is 100% substituted with N1 methyl pseudoadenine for reduce the immunogenicity and toxicity. So, GenScript ready to use IVT mRNA reagents are characterized by their reliable performance. And here I shown the expression of ESP Cas9 mRNA in A549 cell was evaluated for 48 hours and post transfection using ELISA and GenScript. ESP-Cas9 mRNA exhibits a superior performance compared to the competitor product. Not only that, but we also providing the ES mRNA into the nanoparticle formulation. So here the study is carried out by two different LNP formulation are generated. One is ALC0315 and LP01, and 2.4 microgram of each are LNP formulation were incubated with uh, um, HEK cell and Jakarta cell. And cells were lies at day 2 and day 3 for PCR and Sanger sequencing, and the gene editing efficiency data you analyze using the IC analyzer tools and GenScript software. And we can see the LNP formulation ALC0315 shows 80% editing efficiency, more than 80% in 48 hours in both the cell type. And hence, LNP formulation is also important while you selecting to work with the Cas9 mRNA based gene editing. So a common mRNA product will include the essential component is cap, UTR region, ORF region, again UTR region, and then polytail. So mRNA synthesis is multi-step process, and before sourcing the high quality modified mRNA, uh, small or large scale, crucial factor like plasmid and DNA integrity, poly-A tail quality, mRNA capping capacity, uridine modification, impurities, lipid nanoparticle packaging, and manufacturing workflow should consider to get the quality product at the end, because the all these factors affect the end product quality. yield and expression efficiency or translation efficiency, stability, and toxicity. So mRNA synthesis is already, we know, that multi-step process, and to avoid the risk associated uh, engaging a multiple supplier for different step, we understand the requirement of mRNA need one stop solution. So GenScript has developed an integrated manufacturing workflow which covered mm, a wide range of service and QC analysis, and our workflow include from codon optimization to the gene synthesis, cloning and plasmid preparation, then mRNA and circular mRNA synthesis, and lipid nanoparticle delivery system. And GenScript can offer two grade mRNA, RUO and preclinical, and the scale is ranging from zero point 0.2 to 200 milligram. And there are three different capping forms available: cap 0, cap 1, and cap 2. And the default length of poly A tail is 100, but it can be customized to 120. In addition, GenScript also provide three different nucleotide modification. And the recent research shows that the circular mRNA is also becoming more popular. So for that, we also provide the circular mRNA synthesis service, and in addition to linear mRNA. And our LNP formulation service also offer the four kind of formulation, uh, and which allow to select. And our capability for LNP service can also load mRNA size from 20 nt long to 10 kb mRNA, and we are able to produce the LNP from zero point 0.2 milligram to 30 milligram capacity. So, like, if you're working with the cell and gene therapy with the mRNA based process, then definitely GenScript is one stop where you can get all the solution in from one shop. And our customer always receive premium quality mRNA, and these are the some customer feedback, and they are very happy. They even use from different application, in vitro, in vivo application. So we are happy to provide you more information and serve the mRNA services. Another one is the neoantigen directed T cell therapy is getting more popular in nowadays, and as the neoantigen are specific to tumor cell, they are safe targets, and when used in tumor immunotherapy. So basic procedure of neoantigen reactive T cell therapy is shown here, and the first step is involve the neoantigen identification and validation. And for that, like, patient cells are collected, both maybe tumor and normal cell, and then subjected to DNA sequencing and RNA sequencing, and subsequently identify the neoantigen which associated with that one. And at once the neoantigen identify, it's required the neoantigen peptide synthesis. And the second step include the isolation of cell from either from the patient, and then T cell are co-cultured with the neoantigen presenting cells. And after in vitro modification and expansion, the antigen reactive T cell, if needed, it's sorted and can be infused back to the patients. In short, this process of manufacturing of NRT cell, neoantigen based T cell therapy, required the identification and synthesis of neoantigen. So, as the identification of neoantigen is very crucial, to support that work, we are providing the peptide library, and this peptide library can aid in enhancing the process of identification. For example, we are providing T cell truncated library, which is a valuable tool for identifying the specific epitope. Once epitope is identified, this sequence can be used to engineering a personalized T cell for T cell therapy. And to meet the different purity demand for multiple application, we also developed the two type of library. One is the purified peptide library, another one is a crude peptide library, and you can select which one is working best for you initial stage. And also we are offering the bioinformatic tools for creating the diverse peptide library. So you can explore our peptide library design tools to select the most suitable library for your specific application. And peptide synthesis for new antigen is an important part in the new antigen based cell therapy process. However, new antigen peptides are commonly quite difficult to be synthesized as they can be highly hydrophobic and uh, very in the length and the charge and have the strong tendency to aggregates. Therefore, neoantigen peptide synthesis can be significantly bottleneck in the timeline of clinician generating the personalized immunotherapy. In order to help them, we also provide the stage appropriate GM neoantigen synthesis service, and our NeoPrep tap prep platform help to identify the synthetic difficulty and recommend the best synthetic technology use for that specific peptide. So as a GenScript, we have the expertise and technical capability to synthesize challenged neoantigen peptide in large quantity, and be able to provide customized optimizations tool for your downstream application. And a custom peptide synthesis package also option cover like fast peptide synthesis or rush peptide synthesis and regular peptide synthesis, along with the addition of the modification and QC testing service also available. So, with over 20 decades of expertise in the custom peptide synthesis service, we specialize in supplying neoantigen peptide for personalized therapy. And our facility and experienced chemists have supplied over 6,000 custom peptide to over 10,000 scientists worldwide. And our newly established GMP compliant manufacturing allows us to offer comprehensive one-stop service from peptide development to IND filing to clinical trial. And by understanding the unique nature of each project, our team can collaborate closely with customer to the deliver uh, custom specific solution according to their need. So here are some customer which already use and they in the different kind of our services. One of them is 3T Biosciences, which collaborated with the GenScript since 2017 and producing thousands of peptide, aiming to understand how T cell receptors recognize their target. And with this information, they can better identify new target in solid tumor and create the safe and efficient T cell therapy. Another one is the Avidia therapeutic, where scientists screen hundreds of neoantigen in mice to confirm the generalized SNP7 slash 8A, and this significant task was made possible by um, working with the GenScript peptide chemist who rapidly synthesize even the most challenging sequence to meet their requirements with high purity. So they are also very happy to work with our peptide teams. And now, after talking about the non-viral cell engineering platform, here I would like to give some overview about the viral vector-based cell therapy and how GenScript can support the viral vector-based cell therapy also, because certain customer also prefer to grow viral vector or combination of Cas9 and the LVV vector together. So we are providing different kind of services for them. So GenScript provide high quality, ready to use LVV virus vector design for in vitro application. And these lentil viral vector LVV are created using the range of exclusive technique and material that have significantly enhanced the production process for third generation of lentiviral. So we are providing third generation of LVV, which shows high efficiency, high titer, high safety, and high purity, and which can be provide the superior transduction performance. And we, along with that, we're providing microplasma testing and bacteria free, and confirming that there is a no trace bacteria there, and it's also serum free. So here we shows like we have capacity to go from gene to lentivirus, one-stop packaging service. So once you submit your gene, we can provide you LVV packaging service. And GenScript also provide the different quality of plasmid, because plasmid is also one of the important tools maybe customer using at different point in their workflow. So we offering customize customer the flexible plasmid solution, maybe RUO, or we also providing preclinical grade. And this preclinical plasmid service include the various scale from microgram to 10 gram level, and we follow the strict default QC standard and provide the comprehensive add-on QC option also. So our key service include like highly customizable suitable plasmid with ITR integrated for AV production and AV ITR guarantee also. So along with LVB also provide the AV virus for the research purpose. And uh, if you're looking for clinical grade plasmid, and the our ProBio division, which is a CDMO service provider, also providing these with different scale, and they are capable to provide manufacturing facility even in they expand the manufacturing facility in United States, and they have the existing facility in China also. So GenScript ProBio having a successful completed over 300 clinical batches and more than 20 CMC projects associated with plasmid production. And so once you use the REO grade plasmid, and if you want to transfer that project to clinical grade, definitely the ProBio division can help you to do that one. And GenScript ProBio is also a comprehensive CDSO service provider, as I mentioned earlier. So they are able to provide the all different kind of vector service like LVV and AAV, and they are developing all different kind and different scale, and even GenScript ProBio support the developing both adherent and suspension platform to develop and manufacture LVB virus. And AV vector also we providing where it's and it's also helping them to file the IND application for clinical trial. So in general, like you can get all different kind of cell engineering services from the GenScript, either it's non-viral or viral. So I like to say that our cell therapy products and custom solution are crafted to meet unique requirement, and it's also support your clinical manufacturing needs and allow you to smooth transition from discovery to clinical application with certainty or with confidence. And the stage appropriate solution help you work on whatever you need now and in future. So we are here for you at every steps and assisting with the technical matters, ensuring top quality and providing the regulatory support to your needs, and we help you smoothly progress towards your goal. So that's include our cutting edge solution, customization solution, quality assurance, and our collaboration and commitment to advancement will help you definitely to bring your product in market. So GenScript Life Science is providing not only in cell therapy solution, but we are providing comprehensive solution in any application segment, including gene therapy, cell therapy, antibody therapy, vaccine, and IVD section. So at the end, I like to say thank you to everyone to join this webinar. And due to the time limitation, maybe we're not able to take your question. But if you have any question and any more detailed information you are looking for, do not hesitate to contact us. We are always here to listen the customer's need and always helping the customer and make sure they receive what they need. So thank you everyone to joining this webinar.
Thank you again, Daksha Patel, for your time today and your important research. Be sure to also check out GenScript's booth located in the exhibit hall for more great information. Now, before we go, I'd like to thank the audience for joining us today, and I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 3rd, 2025. LabRoots will alert you via email when it's available for replay. So we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, goodbye.